Good afternoon, everyone. It's my great pleasure to extend a warm welcome to all the distinguished guests, faculty members, researchers, and students who have joined us today for this ANRF Pair webinar series. Today's lecture is organized as part of the ANRF project at Mahatma Gandhi University titled AI Integrated Risk Factor Prediction, Biomarker Development, and Advanced Diagnostic for Metabolic Dysfunction Associated Steatotic Liver Disease. It is indeed a privilege to welcome our esteemed resource person, Professor Dr. S. Anas, a distinguished scientist and academician from the School of Chemical Sciences, Mahatma Gandhi University, Kottayam, for the presentation entitled Innovative Synthetic Strategies Towards Therapeutics for Fatty Liver and Diabetes. Professor Anas currently serves as the Honorary Director of the Advanced Molecular Materials Research Center and Joint Director of the Institute of Integrated Programs and Research in Basic Sciences at Mahatma Gandhi University. His academic leadership has also been reflected through his service as a member of University Senate from 2018 to 2023 and as a member of Board of Studies in several autonomous colleges and universities. Professor Anas earned his PhD in Organic Chemistry from CSER NIAST, Thiruvananthapuram in 2008. Following his doctoral studies, he pursued prestigious postdoctoral research at the University of Paris, France, and later at Uppsala University, Sweden, gaining valuable international research experiences. Professor Anas has made significant contributions to chemical sciences through an impressive research portfolio. He has published more than 57 research papers in reputed international journals, edited one Elsevier book, authored 6 book chapters, published 65 conference proceedings, and has been granted one Indian patent. He has delivered more than 63 invited lectures at national and international conferences, reflecting his recognition as an expert in this field. His research achievements are highlighted by an H-index of 20, I-10 index of 36, and over 1,400 citations. As a dedicated mentor, he has successfully guided 8 PhD scholars, 1 postdoc fellow, 6 MPhil students, and more than 70 master students, thereby contributing significantly to the development of young researchers. And Sir also associates with several journals as the editorial board member. In recognition of his outstanding contribution to chemical sciences, he has been recently honored with the prestigious Kairali Research Award 2024 by the Government of Kerala. His current research interests include transition metal catalyzed organic reactions, polymer supported catalysis, and polymer nanocomposites, areas that are at the forefront of sustainable chemistry, advanced functional materials, and industrial innovation. So, dear participants, today's session offers us an excellent opportunity to learn from a scientist who works seamlessly, integrates fundamental chemistry with cutting-edge applications. On behalf of the ANRF Pair Project, Mahatma Gandhi University, I extend a heartfelt welcome to you, sir, and we eagerly look forward to your enlightening session. I proudly acknowledge that Anas sir is one of the co-PI and joint project director of our ANRF Pair Project at MG University. Once again, I welcome our Professor Dr. Anas and all the participants for today's webinar. I wish everyone a very informative and enriching webinar. Over to you, sir. Thank you.
Yeah, thank you very much, Dr. Chandana, for the kind words of introduction. As rightly pointed out here, today I would like to give a webinar on the topic which is related to the NRF project entitled as Innovative Synthetic Strategies Towards the Therapeutics for MASLD. So, as rightly pointed out here, NG University has got this NRF project under the sub-theme metabolic diseases, and our MG University entire project is entitled AI Integrated Risk Factor Prediction, Biomarker Development, and Advanced Diagnostic for Fatty Liver and Diabetics. As part of this, we here working 9 project investigators, and I am one of the project investigator working under the work package drug development, and especially we are trying to target a therapeutic compound for liver regeneration in fatty liver. And my group or our sub-project main objective is to design or identify novel cost-effective synthetic protocol for the development of drug for the MASLD. As you may be knowing that this MASLD is one of the most common chronic liver diseases affecting 1/3 of the adult globally. So people are trying to develop various methodologies, various approaches to prevent this thing. And especially the problem is that the MASLD prevalence is rising rapidly alongside the obesity and the type 2 diabetics. Mainly it is associated with the lifestyle diseases, and people are very much concerned about things. So we have to be very careful in understanding and finding out the solution for this particular disease. So as part of that, we know that I'm directly entering into my project and what we are going to do. Especially for a disease, people firstly we think about the drugs for these particular diseases. So these are some of the major drugs so far used for the MASLD. So in this list, you can see that the only exclusive medication for MASLD is the resmetirom. This is one of the this is commonly tradenamed as Resdifra, and this is one of the important drug which is prescribed for this MASLD exclusively. Along with that, there are some other medicines available in the market. I have listed here, and it is anti-diabetic and anti-obesity agents, and also some other drugs are in pipeline and clinical trials. So it is very important to develop a drug for a disease. So this is my major objective. So we have selected resmetirom as the key therapeutic agent. As I have mentioned that this is a one of the important and exclusive drug for MASLD. Well, how it is acting is it is considered as a potent and highly selective thyroid hormone receptor agonist in clinical trials. So the beneficial effect of this TH or thyroid hormone on lipid levels are primarily due to its action at the thyroid hormone receptor beta receptor THR beta in the liver. So while using this kind of THR receptor, there may be some adverse effect, including the cardiac factors, which are mediated by the influence of or interference of the THR alpha. So people are, especially when thinking about the MASLD, trying to develop some THR beta receptor which is very active for this kind of MASLD and should not affect the should not have this side effect of the THR beta. So this is our main focus area. So this is the structure of the resmetirom. I am, as rightly pointed out here, my expertise in synthetic organic chemistry. We are synthesizing the molecule, so we are very much concerned about the structure of the molecule. This is the structure of the resmetirom, and it is the first FDA approved drug for this MASLD, and it is a THR beta agonist, and it has got a direct effect on liver fat reduction. So that is why we have selected this molecule for our study. And based on this, our work plan can be broadly summarized as it aims to design and develop novel synthetic strategies towards the synthesis of this molecule. So there are so many methods are available for the synthesis. I will be discussing some of the methods in the coming slides. So in the identification of this particular drug, we are mainly concentrating on two approaches. One is that is the primary thing. We are going to develop, or we are planning to develop a novel and eco-friendly sustainable synthetic route for this particular molecule. How we can achieve this molecule in simple step or a sustainable route. That is the first approach. And second approach is we are going to evaluate the effect of or consequence of the synthetic modification of this particular molecule, and how this modification influence the activity, and that is how we are targeting novel drug molecule. These are the main broad approaches what we are trying to do. So this can be summarized in the overall work pattern can be summarized in this flowchart. So we have the drug molecules. Here we have selected for the primary purpose. We have selected the resveratrol as the drug molecule. And as I mentioned earlier, we are going to, or we are trying to develop a novel synthetic route for this synthetic synthesis of this molecule. And here we have to follow the retrosynthetic analysis. So we will be preparing or identifying some of the novel routes, and based on that, we will synthesize the molecules in a cost-effective and more importantly, sustainable manner. This is the first approach. And second approach is we have to find out the development of novel drug molecule. It is somewhat tedious task, but how we can achieve this is, before going to the actual synthesis, we have to do so many computational analysis, in silico analysis, and where we will modify several functional groups. And this work is almost progressing in a good manner. And after that, once we are identifying one or two key molecules after the analysis, we will try to do more computation studies, molecular stimulation, molecular dynamics and docking, and further some DFT analysis, etc., to fine-tune the most suitable molecule for our target. And finally, we will synthesize that molecule in the lab. And this is how the synthetic plan is there. Of course, after the synthesizing a novel molecule, we have to do a lot of in vitro analysis and further biological analysis, etc. That is not included in this part of the lecture. So this is the main approach we are planning to follow up. So this is the first part, the development of the novel synthetic route. So as all of you know that the traditional therapeutic routes for any molecule usually involves the multi-step pathways. So many steps we have to do. So if we can able to reduce the number of steps, or if we can contribute in any one step by imparting the green chemistry or greener routes, that would be highly useful. So it is very challenging to develop reduce the number of steps for the synthesis of the molecule. So traditional therapeutic routes suffer from multi-step pathways, high reagent footprint, heavy cost, suboptimal reaction yields, and poor atom economy. Therefore, it is very, nowadays, people are research focus concentrating on transitioning targeted chemistry toward the sustainable green route. So many routes are available. Green chemistry is a vaster area. So we will adopt a maximum greener aspect into the synthetic protocol. So in this aspect, we have to look how the resveratrol is synthesized so far. That is very important. We looked into the literature, and it was interesting to find that one of the successful report on the synthesis of the resveratrol was reported in 2007 and patented in that year by Madrid Pharmaceuticals. That was the first report, and later some other reports are also came into literature. I am explaining one by one most important things. This is the structure of the molecule. So this is the classical first synthetic route for this molecule. You can see that those who are well aware in the organic synthesis maybe see that we have to follow 7 steps, especially using some of the cost reagents and some harsh reaction conditions, etc. Anyhow, this was patented by the Nancy Ellen et al. In the 2007 for the synthesis of this resveratrol, and this was the first report. And later, the same group and same company improved these protocols in 2014 by, even though it was containing the same, almost the same number of steps, similar number of steps, they have replaced some of the toxic or some of the costly reagents with the more cost-effective and some comparatively greener conditions. And this is other important route available in the literature. It was published in the Journal of Medicinal Chemistry after the patent again in 2014. So in between, there were not much report or effort to synthesize this molecule. And after that, several reports are also available, but that were not much important, or in terms of the step or greener aspect, it was not so important. And one of the major landmark in this area came recently, especially after starting our project in the year 2025 September. After starting our project, there was a challenging synthesis they have done by the Zai and Covercates, and here they have followed only 4 steps. starting from dichloroperidazine, and they synthesize the molecule in 4 steps. So this is the most cheaper and easiest method available for the synthesis of this molecule. In this context, we are planning to develop, or we are planning to contribute to novel synthetic route for this molecule, comparatively cheaper and easier than this. So this is the first approach. You can see that I can see an example. This is the proposed route from our side. This is the retrosynthetic analysis of this molecule. We are planning to synthesize this resimetron by the coupling between these 2 substituents, and which could be synthesized by this molecule azauracil and this difuran 2,5-dione, furan difuran, etc. This is a retrosynthetic initially we are proposing, and based on this is a synthetic plan, that is a retrosynthetic analysis. Based on this synthetic plan, this is the synthetic plan we have developed. Here we can synthesize the resimetron in a 4 step, especially by coupling between these 2 components, and this can be synthesized from the isopropyl furan 2,5-dione in single step, and this component can be synthesized from the azauracil in 2 steps. And this is the synthetic plan we are proposing. We have started some initial experiment on this, and not much progress has been obtained in this particular part of the project. It will be done in the coming months and coming years. This is the plan for us. If it is succeeding, it would be the most easiest and shortest route for resimetron. This is the plan A, or the first part of our work. And second part of the work, we are planning to develop novel drug molecules. As I mentioned earlier, this is very important. So before proceeding to our work and results, I would like to give a fundamental understanding. Maybe most of you are aware. Those who are not aware, I just give a fundamental understanding how a drug is acting with the receptor. If you have a drug and it will be interacting with the receptor and leading to the drug receptor complex. Usually the receptor can be the protein or receptor, etc. So it is very important. Please note that it is very important to have the suitable or correct structure of the molecule which fit with the structure of the receptor. Then only we will get the positive results. This is the fundamental of this interaction of the drug with the receptor. Okay. And coming to the resimetron, how this drug is interacting with the receptors. So recent studies revealed that how resimetron interact with the receptors. For example, a THR alpha and THR beta, the thyroid hormone receptor alpha and THR beta. So many studies have been reported in recent years. So we have to understand how this molecule is interacting with this, how the structural modification occurs in the resimetron is making any changes in the mode of interaction or it binding energy, etc., etc. That is the more important thing. And based on that, we have planned. So before proceeding further, I would like to give a fundamental idea about how these THR alpha and beta are functioning in the body. For THR alpha, it may be noted that its primary location, these receptor locations are in bone, skeleton, cardiac muscle, etc. At the same, but in the THR beta is in the liver, kidney, etc., etc. That is why the MASLD is very active for THR beta, which is connected with the liver. So this is the problem. That is why we have to be very careful while designing a drug for MASLD, because this is a THR beta agonist. That is why it has to interact very nicely with this THR beta. The drug has to interact very nicely with the THR beta. At the same time, it should not interact much with the THR alpha, because if we have more interaction and we are more binding, that may adversely affect the problem, may lead to the cardiac problems, etc. That was the thing I have mentioned earlier. So we are trying to find out a drug or molecule which is having a very good binding with the THR beta and less binding with the THR alpha. That is the fundamental target or fundamental idea of designing this new drug candidate. Okay, for example, this is the resveratrol molecule, and this is the receptor THR beta. So if we are taking this medicine, it will go to the binding pocket of this protein or this receptor in the suitable manner. How effectively it is coordinating is significantly influence the outcome of the drug action. Okay, so now what we are trying to do is we are trying to modify the structure of the resveratrol. So any modifications in the structure of the drug molecule significantly influence the interaction with the receptor. That is the hypothesis. Okay, so let us see how it has been influencing. So in this aspect, the overview of our current work is that this is the structure of the resveratrol. We will do some of the functional modification or structural modification of this molecule targeting the single drug molecule, and for that we are doing the computational analysis, in silico analysis through the docking studies, ADMT, ADMET, molecular dynamics, DFT, etc. Here we have completed the docking studies. I will be giving some glimpses of the result of the docking studies. This is the overview of the current work in search for the target molecule. So we can see, please look at the structure of this molecule. The first one is the resveratrol reference molecule, and now we are screening, we are giving the result of the docking studies. You can see that the several functional group modifications we have done. Here we are trying to modify the effect of the linker unit. Actually, it was oxygen. When it was replaced with the sulfur, methylene group, carbonyl group, amino substitution, etc., etc. And we have done the corresponding docking studies and found that the result and somewhat positive results were obtained, even though it was not significant. You can see that the docking score of the when we are replacing oxygen with the methylene group, the docking score has been improved, and comparatively better results obtained with the carbonyl group. You can see that THR beta is showing the better interaction, and THR alpha is going lower interaction than the reference molecule. So our idea is to select a molecule, modified molecule, which is having better docking score than the THR beta and lower docking score in the THR alpha. Then only we can target the molecule. Okay, so I am going to show the complete work now. Several modification. Another modification we have carried out in another functional unit. You can see that is highlighted in yellow color. Several modifications we tested, and the corresponding the results are shown here. Here also we can see that the significant improvement in the docking score, especially when we are using the aldehyde, allyl group and methyl group. And more interestingly, you can see that when there is hydrogen, that means there are no other substitution. The actual place it was nitrile group. When there were no nitrile group, you can see that it's class tremendous increase from 9.6 to 12.1 in the magnitude of this particular binding energy value. That is very promising and hopeful. And based on, we have not stopped here. We have carried out the several other modifications. We have tested the entire entire this ring part has been changed. This is the several trial. These modifications are based on some of the existing report, how this particular functionality influenced the result, and the corresponding docking scores are here. For the time constraint, I'm not going to detail the one by one results. Anyway, even though it is a slight change, there are some changes are happening in the docking score, especially in the THR beta. This is the last set of molecule which we have tried, especially we are in some cases we have completely avoided the ring and open structure we have tried, and here also the docking score has been changed, and these are the some of the results obtained. Here the red colored things which I highlighted is better than the reference molecule resveratrol. So based on that, this is the final set of how the stereochemistry of a molecule, the R and S isomer of a single molecule, whether it is influencing that, but that has not much influence on the stereochemistry. Okay, that is just a study to analysis the stereochemistry or the chirality of a carbon, how it is influencing. So based on this total, I can summarize that we have now almost screened 53 molecules by changing the functional group or in the resveratrol molecules, among which the 29 molecules have been selected for the further studies, because that was the criteria was better than the resveratrol docking score. So it is only based on the docking studies. So we have now selected the 29 molecules. These are the 29 molecules and the reference resveratrol which we have selected. We cannot proceed with this entire 29 molecules. We have to refine and filter into 1 or 2 molecules. That is why we have to do the further studies. So anyhow, based on this initial docking studies, we have now identified the 29 potent drug molecules, and further studies have to be done. Now we have selected the 29 molecules, and we have to study how it is affecting the absorption, metabolism, excretion, and toxicity, etc. Based on that only, we can further molecular dynamics and DFT studies to understand the stability, etc. So 29 molecule can be among the 29 molecule, we will find out 1 or 2 hit molecules, and that molecule will be selected for the synthesis. And this is the current situation right now. And we hope to develop a novel drug molecule through this research. And thank you. And I hope I have given a broad overview of the work which we have done. I'm very happy to answer any queries related to that. Thank you. Thank you very much. Chandana, can you hear me?
Thank you, sir, for the valuable and informative session. Now the session is open for discussion.
Hello. Chandana, hello. Can you hear me?
Yes, sir. Yeah. Thank you, sir. Thank you for
I'm not hearing.
Hello. Hello. Hello.
Hello.
हाँ सर. Hello. Vaishnav, am I audible?
No, കേൾക്കുന്നില്ല.
You just conclude the session, okay?
Okay. Good afternoon, everyone. On behalf of ANRF Pair Project and myself, I extend my heartfelt thanks to our esteemed speaker, Dr. S. Anas, for sharing such valuable insights and expertise with us today. The engaging presentation and thoughtful responses have made this webinar both informative and inspiring. I also would like to extend my sincere gratitude to all the participants to your active involvement. A special word of appreciation to the organizing committee and technical support for having a smooth session. We hope today's session was beneficial and that you will continue to join us for future programs also. Once again, thanking everybody for attending. Thank you.
Yeah, thank you very much. Have a nice day.
Thank you, sir. Thanks.